Hi, everyone. Thank you all so much for coming today, and for those of you who are joining us online as well. My name is Michelle Rangarajan. I'm a physician scientist based primarily just down the road at Mass General Hospital and also here at the Broad. And I'm also a parent of 2 kids with Duchenne muscular dystrophy. And wearing all 3 of those hats on behalf of both the Ladders to Cures initiative here at the Broad and Parent Project Muscular Dystrophy, I'm thrilled to welcome all of you here today. We're here to talk about how to administer gene therapies to the subset of patients who have pre-existing immunity to the viral vectors necessary for delivery. On the face of it, this phenomenon of adeno-associated virus, or AAV, seropositivity, sounds awfully specific, particularly in this Ladders to Cures series that more recently has included topics like biology in the age of AI or decoding genomes. And yet, as I probably don't need to tell anyone who's here, in this growing era of genetic medicine, where naturally occurring and evolved AAV are the basis of the current and next generation strategies for gene replacement, as well as in vivo gene editing, and where 1/3 to half of otherwise eligible patients with devastating progressive diseases cannot receive these therapies because of pre-existing antibodies from environmental exposure, this specific challenge is clearly a linchpin in achieving success in the broader fight against rare genetic disease. As you will hear about today, solving this challenge means understanding and addressing scientific hurdles that sit at the intersection of AAV virology and immunology, immune modulation, clinical testing and trial design, as well as features that may be specific to certain diseases or certain transgenes. Solving this challenge also likely means identifying ways to make gene therapy safer, potential for repeated dosing, and above all, bringing potentially transformative therapies to many patients who need them now. We are thrilled to have an incredible lineup of speakers today, but we also know that many of you in attendance have insight to add. Perhaps you're a transplant physician who administers the medications we're talking about every day, or an expert in the biology of circulating and tissue plasma cells, or have run AAV clinical trials. Regardless, we would like to hear from you. We've structured today with 2 extensive moderated panel discussions that we hope will include discussion and questions from the audience as well. We hope to dive into details and sometimes conflicting data together and are aiming for a rich and robust discussion. We really hope that such discussion will help us identify an actionable path to address the scientific hurdles that stand in the way of gene therapy for patients who are seropositive. We have mics at the front if you're able to come down to ask questions, or Katie will be running with a handheld mic, so just wave your hand enthusiastically during the question sessions. I also want to take this moment to thank Anna Greca for her leadership of Ladders to Cures and supporting this endeavor, as well as Jillian Shaw and Katie Ligori, without whom none of this would have been possible. We're deeply grateful to Pat Furlong and Kimberly Galbraith at Parent Project Muscular Dystrophy. And of course, we couldn't do any of this without Kevin, Scott, and Sean in the AV booth. We're also deeply grateful to all of our speakers, moderators, and discussants who have traveled from all over the world to be here today. With that, I'm eager to hand over the microphone to Federico Mingozzi, who will chair our first session. Federico is a seminal voice in AAV biology, and particularly AAV immunology, including early involvement in the ocular gene therapy that became Luxturna, the first AAV gene therapy approved in the US. He's also extensively characterized immune responses to AAV vectors in humans and developed strategies for modulating immunity to AAV, including defining work with the IgG cleaving endopeptidase, IDIS, or imlifidase, which we will hear a lot about today. Federico was also incredibly generous with his time and thoughtfulness in helping us put this program together, and so I'm also just deeply grateful for his help. Thank you so much, Federico.
But we're on the front row.
Not from the microphone.
In fact, this is going to be...
You're good? It's on.
Oh, you can hear me now. Okay, great. Well, I'm going to be here only for a couple of minutes, actually. I just want to introduce the first speaker. It's an honor to introduce Dr. Kathy Hai, who's a true pioneer in the field of gene therapy. Kathy did a large number of seminal studies across academia, particularly when she was at Children's Hospital of Philadelphia, University in Pennsylvania, and in industry, Spark Therapeutics, where she was co-founder and head of R&D for several years, and then she was at AskBio. I would say there's a lot that I could tell you. I could speak for hours, but in fact, her work led to the approval of 2 drugs. One is Luxturna for inherited ocular disease, and then Bequez is the recently approved gene therapy for hemophilia B. Her talk today will explore the experience in the clinic with patients with pre-existing immunity to AAV, particularly antibodies to AAV. Kathy, you're here, okay. And I have to go back there.
Okay, well, thank you, Federico. So Federico could have said all those same things about himself. He was obviously key to the approvals of both Luxturna and Bequez and has a long history in the field. So I thought that a useful way to start today's proceedings would just be to tell you a little bit about what we know from trials that have already occurred about administering AAV to people with neutralizing antibodies and how that affects outcome and the level of safety, at least to the point that we understand now, and also just to give an overview of some of the solutions that have been considered for people who don't qualify for the initial studies, and we'll be hearing a lot more about that later today. So these are my disclosures. I think what I want to do before I start is also just note the fact that some pre-existing antibodies occur as a result of wild-type infection, and that these tend to be lower titer than the neutralizing antibodies that occur after somebody has been infused with an AAV vector. So Michelle talked a little bit about that at the beginning, but these are essentially 2 different problems for therapeutics. In other words, readministration to somebody who's already had the vector is a different problem from just somebody who's got naturally occurring, typically lower titer neutralizing antibodies to AAV. So there have been many, many studies looking at the prevalence of neutralizing antibodies following exposure to wild-type virus. And typically, these report a wide range of neutralizing antibodies. And there are a couple of reasons for that. One is that people end up looking at different populations. And the other is that the assays for neutralizing antibodies themselves are non-standardized, and they all have different levels of sensitivity. So these data are from a study that was done by Birgit Reipert and her colleagues at Baxalta in Vienna when they were pursuing a potential gene therapy for hemophilia. And on the left you see, their study of 200 donors, mostly healthy donors with a small hemophilia population from the US. The donors were from Europe and the United States. Let me say a couple of things about their neutralizing antibody assay. These were all validated clinical assays. And the second thing I'll say is that to conduct the assays, they were using vector that is empty capsid-free, meaning that the assays had the sensitivity that you see when there are no empty capsids. So you can see here that among these different populations, there was, in terms of neutralizing antibodies to AAV, 8. The lowest proportion was in a group of healthy donors from the United States, about 32%, and the highest was from a group of healthy donors from Eastern Europe, or about 63%, and that there's a range of titers, as you can see here. But if you take all the people who are negative, plus the ones with titers less than 1 to 20, you can see that most patients have relatively low or absent titers to AAV8. They also did a study looking at cross-reactivity, and were able to show that about a third of the patients are only reactive, a third of the positive patients are only reactive to one serotype. These people tend to have lower titers within the group, but 2/3 of the patients who have positive titers are cross-reacting. And so I think that's consistent with many other studies that have been previously reported. And I highlight Birgit Reipert's because it's a recent study and she's a terrific immunologist. So these, in contrast, are data looking out to 14 years following vector infusion in 3 individuals who were in the first AAV liver trial, which was for hemophilia B. And this was an AAV2 vector. I put up here the ages of the patients when they were infused. And you can see that even 14 years later, these people all have very high titer antibodies to AAV2, and that in general, there's a pretty good degree of cross-reactivity as well. So this is, as I said before, a very different problem from what we are looking at in terms of naturally occurring antibodies. I think it's important to point out that neutralizing antibodies don't seem to prevent transduction when the vector is directly injected into a tissue. This was one of our first trials of AAV, factor IX, injected into skeletal muscle. Despite what I just said about naturally occurring titers tend to be low, our first 3 patients had higher titers using this older neutralizing antibody assay with LAC-Z. The pretreatment titers ranged in the first 3 patients that underwent this procedure from 1 to 10 to 1 to 1,000. But this is the immunohistochemistry here at the bottom, staining for factor IX production in the liver in panel B. Panel A is uninjected muscle as a control. Panel B is immunohistochemistry for factor IX. And you can see that at the muscle biopsy done at one of the sites of injection, there's robust staining for human factor IX in the injected muscle, despite the patient's very high pretreatment neutralizing antibody titer. And the same was true for the subretinal injection in the Luxturna trial. We collected the information on people's pretreatment antibody titers, but it never seemed to make any discernible difference in their outcomes. And there's nothing about pre-treatment neutralizing antibody titers in the Luxturna label. It's not measured, and it seems not to be relevant when you're going into the subretinal space. So obviously the situation we know is quite different for intravascular administration. Either in the first liver trial we were going through the hepatic artery, and I think the same can be said if you're going through the coronary arteries, or just intravenous, intravascular approaches. People had, prior to those to that first liver study, had conducted studies using human IVIG administered to mice to mimic pre-existing neutralizing antibody titers, and we knew that these titers that we could measure in vitro in cellular assays had a correlate in that mouse model that blocked transduction. The problem was that for the first intravenous administered or hepatic artery administered trials, we didn't really know where to set the cutoff. And so for that reason, we accepted patients who had positive titers and just tried to understand from the clinical data what effect they would have. And these are 2 of the patients in a dose from the first liver trial for hemophilia B. 2 × 10⁻¹² vector genomes per kilogram was the dose. The subject on the top, subject E, had a very low pretreatment neutralizing antibody titer to AAV2. Subject F had a considerably higher titer. And the red bar is their factor IX level at the beginning before a cellular immune response took away their factor IX expression. But you can see, obviously, that the lower titer did not block transduction. This patient had a circulating level of factor IX of 10 to 12% for the first 4 weeks after vector administration, whereas the patient with the higher titer essentially never got therapeutic levels of factor IX. So That was the beginning of the understanding of what these levels did. I'm going to say that after this trial, it was a while before the next liver trial began, and the next liver trial for hemophilia B was actually done by Art Nienhuis and Andy Davidoff at St. Jude Children's Research Hospital, and in collaboration with Amit Nathwani at University College London. And actually, while they were preparing their trial, we were doing a lot of back and forth discussion and sharing of data. This was, pre-company era. The first companies had died, the new companies hadn't started. And so we were sharing a lot of data back and forth, and in particular, Fraser Wright and John Gray were comparing and contrasting manufacturing processes and downstream purification processes at the St. Jude facility versus the Children's Hospital of Philadelphia facility. And one of the things they uncovered was that the St. Jude vector had about a 10-fold excess of empty capsids, whereas the vector at CHOP had 0 empty capsids. And so in an effort to try to understand more clearly what effect that might have, Federico Mingozzi and Xavier Angela in our group at Children's Hospital undertook a study where they used again that mouse model that had first been developed by Linda Couto, in which increasing amounts of human IVIG are injected into mice, and then based on the level of IVIG that's injected, the dose that's injected, you get increasing titers, neutralizing antibody titers to AAV. And then studying the effect of gradually increasing titers on vector administration with and without excess empties. And so what you can see here is that for mice with no neutralizing antibody titer, whether you have no empty capsids or a 10-fold excess doesn't make any difference. The mice express what we'll call 100% factor IX from that vector dose, which is a relatively low dose, which heightens, of course, the sensitivity to the neutralizing antibody. But if you give the mice enough IVIG to give them a low titer of neutralizing antibody, when you inject the vector with no empty capsids, you get essentially a greatly diminished level of factor IX, but with a 10-fold excess of empties, you get close to levels that are seen in the absence of any neutralizing antibody. And then if you titer this out, I don't know what happened to this slide. Oh my, the first one is lost there. Okay, well, anyway, you can maybe get the idea from here. But the first one was actually a low titer, a low neutralizing antibody titer of equivalent of neutralizing antibody titer of 1 to 1 to 3. And to overcome that, if you put in a 10-fold excess of empties, you can recover close to the situation with no neutralizing antibody. But as the neutralizing antibody titer goes up, here it's a neutralizing antibody of 1 to 10. Over here it's 1 to 100. And here it's greater than 1 to 3,000. You can see that you need gradually more empties to overcome the effect of the neutralizing antibody. And that if the titer is high enough, you just can't overcome it. And so there are a couple of other points that I want to make about this study. One is that there were similar findings in non-human primates with naturally occurring anti-AAV antibodies. And the second is that there were extensive safety studies in the mice and the non-human primates, and the antibodies didn't seem to make a difference for vector biodistribution. Now this is vector headed to the liver, where most of the vector is going to go anyway. But clinical lab studies, histopathology on all the tissues, and measuring levels of AAV antibody complexes, circulating antibody complexes, did not disclose evidence of any kind of problem that would be a safety problem. And so I think the main message from all of this is just that it's the total particle dose, which could be either fulls or empties, along with the antibody titer. So the balance between the total capsid dose and the antibody, the neutralizing antibody titer, is what determines the outcome here. And here's a study that was recently conducted by Unicure, published last year in the New England Journal. And it looked at, this is actually data from the FDA summary basis for regulatory action, but it groups all the patients in the phase 3 trial for hemogenics, these are all hemophilia B patients, into those with the lower range of titers, a mid-range of titers, and very high titers of neutralizing antibody. And what you can see here is that as the titer increases, the average circulating level of the transgene product declines. Now, what I would say that's important about hemophilia B and the dose used here, both 27% and 42% are therapeutic levels. And actually, we don't have any data that says that 42 is really better than 27. Both of those are clearly much better than 1.5, which would really not be therapeutic for patients with hemophilia. So, but this is, I think, not a situation that obtains everywhere. So fortunately for hemophilia, we have a very wide therapeutic window. If you consider other indications, including Duchenne muscular dystrophy, for example, we don't know for certain that a reduction of this magnitude would not present a problem. So let me just close by saying we're going to hear a lot today about potential solutions for patients with pre-existing antibodies. Essentially, you can divide them into two. You can change the product or you can change the recipient. I've talked to you about increasing the particle dose to overcome pre-existing antibodies. This is likely to work at low titers. It may eventually be prohibitive in terms of cost for very high titers or for other reasons that I think some of the later discussants will expand upon in this symposium, and in particular whether it may change vector biodistribution, for example. Are there ways to alter the vector to avoid pre-existing antibodies? And then, of course, people have explored ways of changing the host. Plasmapheresis has been explored. And you heard Michelle talk about IgG cleaving endopeptidase. And we'll hear more about that later today. And of course, I'm a hematologist, so I have to remind you, of course, hematologists know a lot of ways to get rid of B cells and get rid of plasma cells, either transiently or permanently. And there's so many regimens for doing that that I couldn't list all of them. I will tell you about a recent study from Mass General Hospital in homage to our organizer. It's not one she was involved with, but just, okay. So this is a study that was done for patients with membranous glomerulopathy, which is an anti-IgG disease that results untreated in eventual first proteinuria and eventually renal failure. But it's been treated for a number of years with rituximab, but that doesn't always give a long-term remission. And so the investigators there came up with another regimen that you see is a admittedly harsher one of rituximab, Cytoxan, and prednisone administered over a period of 2 years. But you can see that the offending antibody and indeed all IgG antibodies are rapidly extinguished on this regimen, and they stay extinguished, and that 100% of the patients respond to this. Some take longer than others, and in general, those who respond most quickly are most likely to have a complete and durable response, but anyway, everybody responds. So obviously, this is a setting where, what we're talking about with administering AAV, one would expect that the duration of treatment could be much shorter, and most patients would then recover B cell function. It's a fair question whether such an aggressive regimen would be needed in every situation and whether the risk-benefit analysis would justify it. So I'm just going to stop by saying that pre-existing neutralizing antibodies, even at high titer, don't block direct injection into the tissues, but for intravascular delivery, they clearly reduce or completely block transduction of the target tissue, and that the magnitude of that effect depends upon both the dose of the vector administered and the strength, including the titer and affinity of the neutralizing antibody. that high doses of AAV vector have been safely given to people with high titer neutralizing antibodies, and that there are many strategies for reducing or ablating antibody production, and we're going to hear about a lot of those today. So thank you very much. And are we taking questions or? Okay. Okay.
So we do have time for questions, and we have mics down here, or we can give them to you.
I'll take it off, I guess.
Okay.
Great talk, Kathy. Is this on? Okay, yeah. I'm curious, with all the empty capsid work that was done, was it ever looked at to see what the kinetics of the inhibition was, and whether how fast a neutralizing antibody actually work? Because with some new engineered capsids, like we'll hear about today, I think there's some potential for finding the target very quickly, and I'm wondering how much that might play a role in getting around the neutralizing response.
Well, so I do know of one study that we did looking at that. So this was a study in which we took mice and then injected IVIG at the same time, 1 hour later, 2 hours later, 4 hours later. And what you can see is that if you delay injecting the neutralizing antibody for 4 hours, you get no diminishment of the level of expression. So you remember that study, right? So that's what I know about the kinetics. That's not exactly what you were asking, but it's what I know about the kinetics of neutralizing antibody inhibition.
Any additional questions? Maybe I'll ask you a question, because I think it's important to point out are antibodies, in your opinion, a safety and efficacy matter? I think it's important to frame it also in that way.
Okay, I think that's a really important question. And at least from what we know about targeting the liver, and even targeting these other tissues, like skeletal muscle and the subretinal space, it's not a safety issue. I mean, I don't know whether we're going to hear some more data later today that suggests that the presence of antibodies may, if you're aiming for a different tissue, like skeletal muscle, may really alter vector biodistribution. But as you know from the studies that you did, it didn't seem to alter the biodistribution of the vector when you're directing the vector to the liver. And so, I think it's very important to recognize, at least for most of what I know, it's an efficacy issue. It's not a safety issue.
Any additional questions? All right, I guess we'll have time during the panel. Thank you, Kathy.
Okay, thank you.
And pull up my notes. The next presentation is from Ganot, who's a co-founder and also leads patient advocacy at Solid Biosciences. And he was kind to send me a few words about Solid, which is a life science company focused on advancing portfolio of gene therapy for rare neuromuscular and cardiac diseases. I think it would be great to have her perspective as both a representative of the industry, but also a mother affected by rare disease. And so the title of her presentation is AAV Gene Therapy, Caregiver Perspective.
Good morning. Can you all hear me okay? Brilliant. Thank you so much to Federico, Michelle, Parent Project Muscular Dystrophy, the Broad Institute, and everyone here today, both in person, of course, and online, coming together with a common purpose to tackle such a major scientific challenge. To drive the field forward for patients is essential, and I'm grateful to play even a small part today. As a co-founder and patient advocacy and representative of Solid Biosciences, we are a publicly traded company, so I may be making some forward-looking statements. Each of these beautiful boys and young men have at least two things in common. One is that they all have Duchenne muscular dystrophy, and two, they have all been unable to receive AAV gene therapy due to pre-existing antibodies, including my son. Look at that handsome devil. Let me take a moment to explain why on earth I have the privilege to speak with you today. I'm not a scientist. I am not a medical doctor. This is a scientific meeting, and let's face it, you just heard from Dr. Kathy Hai, who of course is one of the world's leading experts in gene therapy. But today is about collaboration and advancing innovation for the patients who need it most. Patient and caregiver perspectives are critical in this pursuit. Now this I know a whole lot about. In October 2012, my husband, Ilan, and I sat in a fluorescent lit consulting room at the Portland Hospital in London, where we were told our then 2-year-old son, Eitani, has Duchenne muscular dystrophy, a rare, genetic, progressive, life-limiting muscle wasting disease. We learned that he would lose the ability to walk by his early teens and need a power wheelchair. And after we all do our best to adjust to that new non-ambulatory phase, physically and emotionally, with home adaptations, transfer equipment, and a wheelchair-accessible van, the relentless beast that is Duchenne would then rob our son of his upper body strength and the ability to do even the simplest tasks independently. Getting dressed in the morning, brushing his teeth, feeding himself, rolling over in bed, hugging his family, would all become impossible. He would require mechanical ventilation to help him breathe, and eventually, as the heart is a muscle too, he would succumb to cardiac or respiratory failure in his mid-20s. And there are no treatments available to halt the progression of the disease nor cure. How is this possible? I was looking at my perfectly healthy 2-year-old boy. Yes, he had big calf muscles, which we all know is a hallmark of Duchenne muscular dystrophy, and he waddled when he tried to run, but this really threw us for a loop. Before that moment, we had never heard of Duchenne. Growing up, I remembered seeing the MDA telethons with Jerry Lewis, but without a personal connection, I didn't really understand the depths of the challenges Jerry's kids faced. With no family history of muscular dystrophy, we were in shock. The feeling of being totally and utterly powerless to help our son was unbearable. We had to do something. Within a year of Eitani's diagnosis, with the support of the Duchenne community, individuals, investors, mentors, and numerous experts, we moved our family from London to Boston, the biotech capital of the world, to start a company dedicated to developing meaningful treatments for Duchenne. And so, Solid was born. In 2018, we initiated an AAV microdystrophin gene therapy clinical study with our first generation construct, SGT-001. Over the past decade, we have learned so much, both as a company and the field of gene therapy. This work is certainly not for the faint of heart. I often tell families that I think of AAV gene therapy as being more at the stage of the Wright brothers' first airplanes. It's pioneering, groundbreaking, and represents a monumental first step in gene therapy. Just as those initial planes were instrumental in getting humans off the ground, AAV has brought gene therapy to life, helping us deliver treatments to patients. But like those early planes, AAV technology still needs advancements before it reaches the sophistication and precision of modern aircraft. We're in the foundational stages of gene therapy and with exciting potential for next generation technologies to take us even higher. We were hoping Eitani could be an early, brave explorer in gene therapy. In 2019, however, we received another devastating blow. And of course, I don't envy the physician that had to make that call, especially as he's in the second row here today. Eitani's antibody titers were too high, which meant he was ineligible for gene therapy. Despite the pioneering nature of the first generation gene therapies, I think we can all acknowledge the work is not done. The high unmet need remains. Many families in the Duchenne community often say, "We were so grateful our son had the opportunity to receive a gene therapy, but what's next? My son continues to decline. What can we do? Can he receive gene therapy again? We need more, we need better." These families deserve our commitment to advancing solutions that address these challenges. Solid Biosciences has grown significantly in clinical experience, research and development, and manufacturing capabilities. Our work is always informed by and in partnership with the Duchenne community around the globe. Building on our scientific advancements, we've also expanded our focus to include related neuromuscular and cardiac conditions. We are excited to be back in the clinic with our Inspire Duchenne AAV gene therapy clinical study, testing our next generation construct, SGT-003. We are grateful to the courageous patients and families who choose to participate in clinical research, to our investigators, advocacy groups like PPMD, who work alongside us every step of the way. Solid is deeply committed to addressing the immunology challenges associated with AAV delivery. We are pleased to be part of today's discussions and confident that this meeting will spark new ideas, collaboration, and pathways. My colleague Sharon McGonigal will be speaking in the next session on the challenges of models and measurements in AAV antibodies. While I'm speaking about Duchenne today, I know this meeting encompasses many conditions beyond Duchenne. The challenge of seropositivity extends across numerous diseases that could potentially benefit from AAV-delivered therapies. Finding solutions to these immunology barriers is essential not only for boys and young men living with Duchenne like my son, but also for countless patients with other conditions who stand to gain from these advancements. We have a shared responsibility to ensure that no one is left behind as we strive to advance gene therapy for all. On behalf of the Solid Biosciences team and this Duchenne mama, thank you for your time today. I have included my email address if anybody would like to reach out. And I'm happy to take questions. Thank you so much.
So we have, as always, time for questions. Maybe we have one question coming.
Oh, I should be here.
Hi. I think we've all seen a lot of non-clinical data where people have used either IDIS in some of your studies, Federico, or plasmapheresis to reduce these antibody titers in animals with pre-existing immunity. In your view, what has the roadblock been to attempting some of these things in the clinic in patients such as your son that has an antibody titer? What's keeping us from moving that forward into the clinic?
That'll be a wonderful question for Sharon and also Dr. Byrne. I think there's many challenges when we're talking about systemic delivery, being able to get to all the skeletal muscle and cardiac. And it's again, there's a lot of different efforts between our company with Solid and other companies in the space, in Duchenne, AAV gene therapy, Solid, Regenexx, Pfizer, Sarepta. There's a lot of companies in the space that are all trying to solve for this. It's not today, but I think we all have confidence that it is sometime in hopefully the near future.
And maybe I have a follow-up question related to that. You're a parent and you know a lot of the, in your job you see a lot of patients and families, right? What is the educational gap, if any, that you see as the biggest when it comes to these gene therapies, the immune responses, I know particularly the pre-existing immunity is something that I've seen families, for example, going in isolation prior to trials for months and months and months to not get exposed to the virus. What do you see as in terms of like information and good education that could be given to these families that is missing?
I think a wonderful aspect of the Duchenne community is, when I think about some silver linings after this diagnosis, is that we do have really strong informed advocacy. So with PPMD, Cure Duchenne, MDA, Jet Foundation, like across the nation, Advocacy is very strong in terms of educating families and doing their best to make sure that they provide all the necessary resources to families, but it's also a very personal decision. So, and that's a decision that families need to make with their clinicians. There are many families where there are multiple sons, like Michelle, that have multiple sons with Duchenne. And sometimes when boys are going for screening, maybe it's the older brother that is going for screening for a clinical study, sometimes the family try to figure out a solution to isolate and to separate those brothers so that if that child is dosed, that the younger sibling doesn't have that exposure, so that in the future, the younger brother may be eligible for gene therapy. But it's so incredibly hard to do, and it's such a personal decision for all families to make. And there's so many other factors in terms of, can they realistically do that? You know, life is life, right? Like, you got to go to your job, you got to get your kids to school. You know, a lot of families that is really hard for them to do, to potentially isolate. And at the end of the day, we don't always know why. I mean, I don't know why my son's titers were off the charts for every, and we know that there's cross-reactivity for every AAV that we tested. We don't really know that information. So that would also be good to know. But again, it's just very challenging across the board. It's personal, but there is a lot of education that's available to families.
Thank you. There's clearly also a big gap in understanding the why.
Yeah.
And how that would happen, right? Even in the context of being close to a person who was dosed with AAV, whether
Right.
or not there's a true risk. I
Right.
think there's still a gap in
We have
terms of
so much to
understanding.
learn.
Yeah.
We do.
Great.
Thank you so much. Thank
Thank you. So I am the next speaker. So Michelle was kind to introduce me, so we don't need that. What I'm going to try to do in the next couple of minutes is really give you a general overview on AAV immune responses, and with some maybe additional information that will be useful for the rest of the meeting and the rest of the discussion we will have at this meeting. I always use this slide to frame immune responses. Very busy, of course, but I always like to frame immune responses to AAV in a way, in a temporal way, right? With in the middle, you see the moment when we receive an AAV vector. Before, even years before, and we really don't know a lot about it, there's exposure to wild-type AAV, which can result in pre-existing immunity. And pre-existing immunity may not even only result from exposure to the virus, but also sometimes we have pre-existing immunity to the actual gene that we express, our therapeutic gene. And the most relevant aspect of pre-existing immunity, of course, we hear about it, we're going to talk about it a lot, is exclusion of seropositive subjects from enrollment in trials or receiving gene therapies, approved gene therapies in some cases. Right after AAV infusion, we have the innate recognition of the virus, and that's really something that happens pretty quickly, within hours or days after vector infusion, which then leads to the adaptive immune responses, so antibodies to AAV, cytotoxic T cells. I will tell you a little bit about it, and then eventually also the potential for transgene immune responses. These two post-infusion kind of arms of the immunity, they have implication in terms of pretty much safety, durability, but also predictability of outcomes across a given patient population. Last but not least, we already discussed the fact that antibody titers post-infusion prevent vector readministration. Katie went into detail about antibodies pre and post-infusion, so I don't want to spend too much time except to say, yes, in general, if you look at pre-existing immunity to AAV, so the natural immunity, antibody titers tend to be on the low to moderate side. It's relatively less prevalent to find subjects with high titer AAV. This is, of course, very much serotype dependent, so that's not true for all AAV serotypes. And then post-infusion for, and sorry, the slides are a little out of alignment. This data is basically what Katie just showed a little bit earlier. Antibody titers that are induced that are high titer, they're highly cross-reactive, persist over time, and so it's hard to think about vector administration with this kind of immunity. When it comes to antibodies to AAV, I think of course, we're worried about it, right? Because they prevent vector administration, but this is not true for all settings. Katie highlighted the experience in the clinic with intramuscular administration of the vector, where we don't care about AAV titers or subretinal administration. However, coming from the clinic, we know very well that intravenous administration of AAV most of the time really is the setting where we are most exposed to neutralization by antibodies, because there's, of course, the vector is infused directly into the bloodstream. And intravitreal administration is another setting where, at least in the context of VEGF, so AMD-related gene therapies, there is some evidence that there are antibodies that run us into trouble. And then again, as we hear before, there's clearly a dependence on the vector dose and our tolerance for antibodies. I think that's quite important because, of course, if we use low doses of AAV vectors given systemically, then our tolerance for antibody titers may be quite low. But then, of course, if our dose is higher, we have a higher tolerance for a certain level of antibodies up to a certain point. And we heard about this study extensively, so I will not go into detail except saying, yes, if we give more capsid, then you can basically overcome antibodies to AAV, and this is something that we see a lot also in protein replacement therapy, where you have anti-drug antibodies, you just dose over these antibodies. For innate immunity to AAV, again, this is just an overview. I think there are some implications of antibodies and innate immunity to AAV. But this is a diagram that basically shows you a little bit. This is a very simplistic view of innate immunity of AAV. What we learn is that, of course, there's a TLR receptor involved in triggering innate immune responses to AAV, specifically TLR2 for the capsid, and importantly TLR9 when it comes to the vector genome. Again, sorry that the text seems to be changed a little bit. And then even before the vector gets to a cell, we've seen in some cases complement-related toxicities in clinical trials. When it comes to TLR and AAV, I think we learn a lot from the clinic. In particular, we learned that the presence of CpGs in the operating frame of AAV plays an important role on the downstream immune responses to the actual vector. And in this table that was published by Fraser-Wright a few years ago, you can see how across several studies, these are all AAV gene therapy trials for hemophilia B. Those studies highlighted by a star, where the vector contained a higher number of CpG or CG dimers, then the expression was short-lived and there was an immune response detected against the vector. Whereas when in the studies where a vector devoid of CG dimers, the immune responses were still detected in some of the studies, but they were more manageable, leading to long-term transgene expression. For complement, I think we're all aware that especially there's a dose dependence, so high doses of vector given systemically, we can see complement-related toxicities. I think the question here is, we don't see that in every single trial, so the dose dependence is there, but it's not across all trials. And of course, there can be a lot of different factors playing into this. For example, there could be differences in manufacturing methods for the vector used in different trials, and of course also complement is pretty variable, complement levels across patient populations. And what is important to remember is that subclinical complement activation may actually still have a role. So even if we don't see a toxicity like TMA, we should be mindful of the fact that complement is part of the innate recognition of a pathogen and may play a role in vector immunogenicity. This is work from Claudia here in the audience, showing in fact that if you look at complement activation, there is of course a dependence on the presence of antibodies, although we learned that also the activation of complement does not necessarily need antibodies to happen in the clinic at least. And what is important also is that, and this is a fairly important point, that both empty and full capsid can drive complement activation. So I think while we love to have more capsid to decrease, to overcome antibodies to AV, then there's a little bit of a point related to the complement. Again, although we see that there's a big discrepancy across studies, not all studies see complement activation. If we inhibit complement, we do also see a decrease in secretion of production of cytokines upon exposure to AAV in vitro. I try to summarize here a few points about the complement. So both empty and full capsid can trigger complement. Presence of antibody may enhance complement activation, although this is not true all the time, right? Because in fact, 100% of subjects receiving an AAV will develop antibodies within days, right? So the relationships here is to be explored more carefully. The targeting of complement may be a way that we could tackle vector immunogenicity. Clinically, there's many drugs available to target complement, and they've been used actually in the clinic to do so, along with other strategies using, for example, B-cell targeting drugs. Of course, there's always the point that will come back to the end of my presentation, is vector purity an element that we should be thinking about when it comes to complement. For adaptive immunity, again, we're talking about production of high titer antibodies. I will focus on the potential of inflammation, so triggering of T cells. And we've seen in some cases also anti-transgene, not really antibodies, but actually more of a cellular response, for example, in some of the Duchenne's trials, and I'm sure we'll hear about it during today's discussions. Here is a model that we've been working over the years, where we basically, many studies across many postdocs in Kathy's lab led to this model. And again, sorry that for some reason, maybe the transfer of the file scrambled a little bit the words in the slides. Essentially, the model sees the vector coming, infecting target cells, and the transduction of the cell with expression of transgene also is followed by a process of antigen presentation by the actual cell, and that really leads to the flagging of this cell for recognition by the immune system. So many examples of that in the clinic. I try to summarize a little bit what are some of the clinical observations across routes of delivery. This is it's not exhaustive, number one. Second, not everything will follow the mechanism I just showed you, right? Because there's a lot that we're learning about AAV and potential toxicities, even driven by innate sensing of the virus in a cell, which may actually not even require a cytotoxic T cell to drive toxicity. But a lot of the toxicity we've seen were focused on, the liver was one of the main targets of these toxicities, especially when intravenous delivery of the vector happened, but also in some setting where the vector was given in the CSF, where you anyway get systemic exposure. So a lot to take into account. I try to just put general principles here. I will not go into the detail of anything because we will have the chance to do it throughout our day. When it comes to immunomodulation, which has been extensively used in gene therapy, I think these are a few points to bring home. Corticosteroids were the first drug that were used in conjunction with gene therapy, and in particularly in the study led by the team of Natwani Davidov and Art Nienhuis, the hemophilia B trial, which then really led to the use of corticosteroids extensively in many, many different gene therapy trials, although other drugs now have been used, targeting more specifically B and T cells. Ideally, because we are trying to block an immune response that, in the case of the capsids, should be there only for a determined amount of time, the immune suppression should be short and ideally targeted, right, more than a broad immunosuppressive drug. The field in general has a little bit struggled to find a general immunosuppressive regimen, and this is because we're not always treating the same disease. Sometimes we have underlying tissue inflammation, which can exacerbate immune responses. Sometimes the disease would not warrant extensive immunosuppression from a risk-benefit point of view. I already told you about the transient nature of the immunosuppression. Of course, when there's a concern about transgene immunogenicity, this is a different matter. That in some studies, this has been the issue. How long do we keep immunosuppression? And then I think it's important that we think about that one day when we will be able to re-administer AAV, then we'll be in a setting of prime boost. So is there anything we have to be thinking about when it comes to immunomodulation and vector re-administration? This is just an example. This is a study that we conducted of an immune suppressive regimen that we used, which seemed to be working in the context of a gene therapy for Crigler-Najjar syndrome. This is a liver metabolic disease, so the vector was administered at 5e12 vg per kilo, and a regimen based on sirolimus and prednisone seemed to actually be okay and manage liver toxicities, which are shown on the right side of the panel, leading to long-term correction of bilirubin, which is a biomarker of efficacy in this particular disease. And I think we're going to hear about it more sometimes today. I believe this is my last slide. I think it's important to think about immune responses in a way that there's a lot that we can do in terms of vector and what happens around vector administration, vector design, and what happens around vector administration. So there's a few things we'll hear about today. For example, can we actually modify the capsid for immune evasion? There's adjuvant technologies we'll hear about. There's manufacturing that is important to minimize impurities with AAV, associated with AAV. What is also important is that we need to find the sweet spot between having a dose that is efficacious, and we know that higher doses of AAV increase the risk of immunogenicity and potential also the risk of more acute toxicity. So something to keep in mind when it comes to these vectors. And I'm going to stop here. This was just an introduction to set the stage a little bit on the immune response to AAV. Thank you. Any questions?
Thank you, great talk. I have two questions. For escape the side effects of AAV-based gene therapy for seropositive patients, are there any considerations using lipid nanoparticles to package AAV-based
That's an interesting question. So it's the idea of basically packaging an AAV vector into a lipid nanoparticle or a nanoparticle to basically even protect it from antibodies, right? Yes. So I think it's an interesting idea. I think the challenge there, and different groups, and we even explore the idea of providing a bilayer kind of particle in the form of extracellular vesicles to the AAV, and you see some protection from neutralization. It's a process that is not very efficient, also owing to the fact that it's very easy to package a nucleic acid into a nanoparticle due to charge, not so easy to do with a complex protein like AAV. So that has been very difficult to achieve, let's put it this way. But potentially could offer some benefit.
The second question that you talk about immune suppression as a strategy. So how about the utilization of ripamycin or Ripalog for that purpose instead of the corticosteroid?
Yeah, rapamycin or sirolimus has been used extensively. This is your question, right? In the context of AAV trials, alone or in combination with different regimens. And the example I actually showed you, rapamycin was given for several months post AAV infusion. There's many other instances where it was also combined either with corticosteroids but also with rituximab and has shown some efficacy.
Thank you.
Thank you. All right, no more questions. So we're going to move forward with the last speaker of this morning. We have Shari Gordon. She is a senior director of immunology at AskBio. Prior to that, she had a career across academia and industry in institutions like Emory, UPenn, and UNC Chapel Hill and GSK. And so Shari has done also very, very interesting work with the administration of empty capsids to humans and showing what happens to the immune system in that setting. So today she's going to talk about strategies to modulate AAV-specific adaptive immune responses. Thank you.
All right, can everybody hear me? Yeah? All right, first let me start by thanking the organizers for really putting together what I'm expecting to be an incredible workshop. It's already gotten off to a fantastic start with talks from, with the previous talks, as well as to giving me the opportunity to present or work on evaluating strategies to modulate the AAV-specific response. And these are my disclosures. Okay, so we know that AAV is a non-pathogenic, a widely seroprevalent virus, as we've heard already this morning. And so there's a large fraction of the population that have pre-existing antibodies. The graph below, let's see if this is the pointer, there we go. The graph below shows the levels of neutralizing antibodies in 149 healthy volunteers. So these are AAV8 pre-existing neutralizing antibodies. And about 46% of them were seronegative, so they would have been eligible for most systemically administered gene therapy. However, the remaining individuals had varying levels of pre-existing antibodies. And so our goal, certainly, as we're talking about today, is to treat all subjects regardless of their serological status. But one of the concerns with this is if we think that when we are exposed to wild-type AAV, you have this primary response. One of our concerns, from an immunologic perspective, is when we now give our gene therapy to seropositive individuals, are you going to really see a secondary response? Do you see antibodies that are either of higher magnitude or that come up with a faster kinetic? And because we've seen treatment emergent antibodies be associated, as Federico nicely described, in some cases with inflammation, complement activation, and potential adverse events, the question that remains is really what is the risk, both safety and to efficacy, of treating seropositive individuals? Now, when we think about the concern about treating seropositive individuals, it's not just the humoral response. By a lot of work that was done by Kathy and others in the field, Federico, we also know that there are concerns with the other arm of the immune system, the cellular immune response. And we've seen elevated T-cell responses be temporally or generally associated with increased liver enzymes. And so when you're looking to treat now seropositive individuals, one of the concerns is, are you going to have a memory response that's going to expand quickly after dosing? So a couple of years ago now, we did some work with AAV8 empty capsids where we were able to dose healthy volunteers. And shown here is the binding antibodies first from individuals that were dosed with AAV8 empty capsid. Now, we started by screening individuals, and we only enrolled subjects that were screened to be seronegative, and we used an AAV8 neutralizing antibody assay for screening. And not surprisingly, we saw high titer binding antibodies. That was really expected, as well as neutralizing antibodies, not shown here. What was surprising to us was actually the T-cell response, which is not quite showing up. Hopefully you can sort of make that out. So the T-cell response, which is shown here, came up within a week of dosing, and we were surprised at the magnitude of the response. So 1 week after dosing, we saw in 5 of 8 individuals a relatively strong CD8, a relatively strong T-cell response. And these were interferon gamma ELISpots that we were measuring. And when we profiled that response quite carefully, we found that that response in individuals with that high magnitude actually had increased breadth. So the response was targeting multiple epitopes, so that was one. And when we dug a little deeper, several of the individuals with that high magnitude T-cell response we saw actually had low-level pre-existing antibodies to other serotypes, such as AAV2. Surprisingly to us, when we looked at this empty capsid, we saw actually a CD4 tropic response as opposed to a CD8 tropic response. And we did that by comparing interferon gamma ELISpots in whole PBMCs versus those that had been depleted of CD8s, and showing that while the AAV-specific response in CD8 depleted was either similar or diminished, in contrast, for example, a CEFX response, which is often CD8 tropic, was diminished by these evaluation. So overall, we have been aiming to develop immune modulation strategies, and we think it's important that this strategy blunts T-cell activation, specifically blunting CD4 responses, which we hypothesize could suppress both CD8 as well as B-cell responses. So the way we've approached this is a combination strategy. We'll need to remove those pre-existing circulating antibodies, but we also think it's important to use some immune suppressive agent, at least transiently, to suppress that initial T-cell response that may come up after dosing. So our approach to that has been first to test several strategies in mouse models. We've used the RH3233 transgenic capsid, which you can give by intramuscular and will give you both a T and B cell response, or by intravenous dosing, as well as several disease models, including Pompe disease models. So we used this mouse model to test a breadth or a variety of agents, and then the best ones we've moved forward into non-human primate studies, and I'll show you a couple examples of that today. So you're going to hear a lot about many strategies to remove pre-existing antibodies. One that has been studied quite extensively is the use of cleavage enzymes that are able to cut antibodies into their FAB as well as FC fragments. There's been quite a bit of work showing efficacy in preclinical models, and these Hansa's imlifidase is probably the most clinically advanced that is now being tested in at least 2 clinical studies. Some of the challenges with these enzymes is that they're certainly bacterial proteins that are themselves immunogenic. There's concerns about the timing of dosing and circulation of cleaved antibody fragments. However, we now see next generation enzymes that have lower immunogenicity, as well as we're now seeing those that can actually cleave not just IgG, but also IgM. Another area that's gaining popularity is the use of FcRn inhibitors. So we know that antibodies, unlike small molecules, have nice long half-lives because they can bind to the neonatal receptor and be recycled as opposed to being degraded. This class of inhibitors are able to bind to that neonatal receptor and prevent the antibody from binding, so that antibody now gets degraded as opposed to recycled. So what you get is, in general, about an 80% reduction in the levels of circulating antibodies. And we've seen preclinical efficacy in different models as well, and these are now being advanced to clinical studies. So we, like others in the field, went ahead to do some head-to-head studies of these 2 approaches, where we infused IVIG, similar system that Kathy described in her talk earlier today. We infused IVIG at different levels in transgenic mice that have the FcRn, the human FcRn receptor, and we're able to show, based on our timing and looking at kinetics, that we could create mice with levels of circulating antibodies that were low level, similar to what you see in individuals with pre-existing antibodies, or at least low-level pre-existing antibodies. We then designed a study to do some head-to-head comparisons, where we first gave animals IVIG, and then came back and gave either a cleavage enzyme or an FcRn inhibitor the second 24 hours later, and then waited 4 days for the enzyme and the Fab fragments to clear, the FcRn inhibitor to really have a chance to reduce those circulating antibodies, and then challenge the animals with an AAV vector that expresses luciferase. And what we were able to find is, or what we observed, is that we do see, certainly in the control animals that just get AAV, which are in green, you can see that there's high transgene expression. When you administer IVIG, those animals are in blue. We see a dramatic reduction or a full blunting of that transgene expression. And the cleavage enzymes were able to fully restore that, while the FcRn inhibitor, it did restore it, but we got a more variable response. So I think there's a lot of good work that is going into showing that these strategies are truly able to reduce low-level circulating antibodies, but we still have a concern as to whether or not they'll be able to reduce high-level circulating antibodies and truly enable all individuals to be eligible for gene therapy. And for that, they likely may need to be used either in combination with themselves or with other approaches. So switching from removing pre-existing antibodies to suppressing the developing response, as you've already heard, certainly corticosteroids is the agent that has been the most used. However, there's quite a bit of both preclinical and clinical work now that's using several agents. T-cell inhibitors such as tacrolimus, B-cell inhibitors ranging from B-cell signaling inhibitors, but by far the alternative that's been used the most in terms of in the clinic has been sirolimus or rapamycin along with rituximab. So we use that as our comparator because we had a keen interest in evaluating an alternative approach which uses co-stimulatory blockade. And so sirolimus or rapamycin certainly acts by inhibiting signals downstream of the T-cell receptor. And then, of course, anti-CD20 or rituximab binds to CD20, the protein on the surface of B cells, and can remove those B cells from circulation. In contrast, we were comparing that, and this is also sort of mixed up in terms of the graphic, but we were comparing that with CTLA-4 Ig, which is a co-stimulatory blockade. So T cells require both signal 1 and signal 2 for activation. Signal 1, of course, comes through the T cell receptor binding to MHC, while signal 2 comes from co-stimulation. This co-stimulatory blockade, CTLA-4 Ig, binds to B7 or CD80, CD86 on antigen-presenting cells, thus denying that T cell from that signal 2 or activating signal. So you're basically inhibiting T cell activation. There are 2 versions of CTLA-4 Ig that have been approved and have been used in different settings, rheumatoid arthritis or even transplant settings for quite some time. So there's a wealth of knowledge understanding how to use these molecules in the clinic, as well as to manage the potential side effects or concerns that come with the use of these molecules. There's also some preclinical data suggesting efficacy in the context of AAV when it was actually expressed within an AAV particle. Our approach has been to give or to do a head-to-head comparison of rapamycin and anti-CD20, or in one group, or AAV with CTLA-4 Ig in another group, and we've been comparing both this in a mouse model, okay, and then that's cut off, okay, in a mouse model as well as in a non-human primate model. So for both of these, we're giving intravenous dosing, vectors in the range of 1 to 5 E12 VGs per kilogram. And what you can see from in the mouse model, oh, and I should say we're starting treatment before AAV dosing and treating for about 30 days. Then we stop all immune modulation and we have a follow-up. For the mouse, I'm showing you data at day 90. For the non-human primates, I'm showing you data at day 70. We're still analyzing the results from the NHPs. So in the mouse model where we used RH3233, so that immunogenic model, what we saw is that we could get a durable blunting of the AAV T cell response using CTLA-4 Ig, which is shown in green. And while we still saw a significant reduction using rapamycin and anti-CD20, CTLA-4 Ig as a single agent was more effective than the combination at reducing that CD8 T cell response, reducing the neutralizing antibodies, and now more recently in a preliminary study with 10 cynomolgus macaques, where you can see this is the level of neutralizing antibodies in the 2 control animals. In the CTLA-4 Ig group, we saw 2 of 3 macaques that had a good blunting of the neutralizing antibodies, while in the rapamycin and anti-CD20, we saw 1 of 3 that had a reduction. Again, this is testing after we've stopped the immune modulation, so we're looking at durability here. Our next question was, well, if you continued CTLA-4 Ig, could you actually get to a point where you could give a second dose? So for this, we went back to the mouse model, to a very immunogenic model, where we gave a first vector administration of RH3233 that expressed OVA. 30 days later, we gave a second administration of RH3233 that expressed luciferase. And we've got control animals that just get the second vector. We've got the second group that gets both vectors as well as the isotype control. And then, of course, our group that gets CTLA-4 Ig, which is basically having CTLA-4 Ig for the duration of the study. We were also curious to ask the question, does the route of administration matter? So for half of the animals, we dosed both the first and second administration of the vector intravenously. The other half of the animals, we dosed intramuscularly. And just to show you that both groups, both the intramuscular and intravenously dosed group, had similar levels of circulating CTLA-4 Ig. And so this table is just showing you a summary first of the antibody response. I'm showing you a snapshot here with samples taken hours before that second dose. And what you can see, and we're measuring both Rh3233 specific IgG, IgM, as well as neutralizing antibodies. And so CTLA-4 Ig was able to significantly reduce the levels of circulating neutralizing antibodies, consistent with a significant reduction in the levels of IgG, but was a lot less effective at blunting IgM, which you would sort of expect based on the mechanism of CTLA-4 Ig. And then when we looked at transgene expression after that second dose, so in this case redosing, we were really intrigued to find that for vectors that were dosed IV, we were not able to obtain a second dose. So you see no transgene expression in the IV dosed group. However, in the intramuscularly dosed group, you are able to see some expression. So confirming and extending what Kathy also showed and discussed earlier is that the route of administration matters. If you're looking at seropositive individuals, you can use immune suppression or other strategies to reduce those titers, but whether or not you're going to get that either dosing, if it's a case of seropositive, or redosing is going to really be dependent on the strategy that's used, the route of administration, as well as your vector titer. So in conclusion, we agree that the immune response to AAV certainly may pose a risk to both the safety as well as the efficacy. However, there are more and more strategies are coming online that can both remove those pre-existing antibodies as well as suppress the developing immune response. We would recommend that these strategies be considered in combination. Certainly cleavage enzymes and FcRn inhibitors are getting a lot of traction and promise in the field. And from our work in both now mouse as well as monkey, we would suggest that co-stimulatory blockade with CTLA-4 Ig may be another strategy to consider when looking to advance these strategies to the clinic. And with that, I'll thank the many people, including an incredible preclinical immunology team. A lot of this work was done by Audrey Fernandez in conjunction with our product development team that does a lot of the mouse work, and an incredible team of supporters and advisors that have been a part of this work. And of course, thank you for your attention.
Hi, Shari. Is it on?
Hi.
Yeah.
Yep.
Really, really spectacular talk. Thank you. In your empty capsid human study, did you administer any immunomodulation for that study, like steroids or?
So that study was done in collaboration with Selecta, and so it was a test of their immune modulator, which was called ImmTOR, which is nanoparticles that contain rapamycin. Mm-hmm. So I didn't show any of the individuals that had immune modulation. I only showed the empty capsid healthy volunteers, so those that got empty capsid and saline. That said, because we were concerned about infusion reactions from the ImmTOR, we did give a single day. So we gave Benadryl hours before, and we gave 24 hours a single dose of prednisone. So they got one dose the day, which was really more to cover, to prevent any sort of infusion-related reaction from the immune modulator, but we gave the controls as well, so it would be a balanced study. So technically they got one day, but the likelihood for that to have a prolonged impact is pretty low.
Okay, and then, thank you, that's really interesting. My second question is, what were the safety profiles in the healthy volunteer study? Did you see ALTs? Did you do like reactive steroids in any way to modulate?
We didn't need to use reactive steroids at all. We did see a few individuals that did have transient asymptomatic liver elevations. Those came up either after the peak of the response, so somewhere around 2 weeks, or we even had one individual that was similar to what you see in AAV, where it came up later in that 6 to 8 weeks. But they were all, again, transient, self-resolving, not needing steroids or any other treatment to lower them.
Thank you.
Can I ask you just anything related to the fact that you use empty capsids versus full that may skew the response towards CD4 versus CD8?
Yeah, so I mean, as a viral immunologist, I always think of AAV sort of as protein shell, and then you've got DNA impurities, other as adjuvants. And I think the nature of the response is going to be determined by, you know, what the vector is composed of. So not having DNA and CPGs may have led to a skewing towards CD4, as well as overall, you know, the characteristics of the host. Is it an inflammatory profile that you're doing? So there are a lot of things. Right. It's also true that, you know, there's been discussions of empty capsids instead of being shuttled to the nucleus the way full capsids are. They may be more likely to stay in the cytoplasm, be proteasome degraded and presented. So the kinetics and the nature of it could be different. I mean, I don't really have a way to show that. We were certainly surprised by both the magnitude and the skewing of the response to CD4. I would not want to say that all gene therapy responses are going to be CD4, but as empty capsids are often a component, I think it's important for us to not assume when you see an interferon gamma response that it's CD8, but instead to actually measure whether it's CD4 or CD8 or some combination thereof.
Thank you. You have another question?
Thank you, wonderful talk. Besides the CTLA-4, are there any considerations and explorations using other T cells signaling like a TIGIT, LAG3, TIM3, or even PD-1?
Yeah, certainly I think that there is a need to evaluate several strategies to blunt T cell responses, and I personally prefer strategies that have a defined mechanism of action, and I think it's important to test them in AAV models head-to-head. So we certainly are testing and have tested several of them. I can't specifically comment on the ones that you mentioned, but yes, I think that, you know, the way forward is to evaluate these strategies head-to-head and try to move the best ones to the next stage. And certainly, you know, the sort of balance between activation and inhibition of T cells is an important one to explore.
Thank you.
One more question.
Hi, Shari. Great talk as always. I had a question with the NHP data with CTLA-4 Ig. Were those NHPs prophylactically treated with each of the immunomodulation, or was it after AAV or co-administered with AAV?
Yeah, great question. So for the rapamycin and rituximab, we actually started a week before AAV administration. For CTLA-4 Ig, we started a couple days before AAV administration.
And the second question is, what was the dose? Was it mid-range, high-range? I know you kind of...
I know. My IP group made me take the doses off, but we can talk about it after.
Thank you.
All right. Thank you, Shari.
Thank you.
This is great.